that there are animals which give birth to young ones and also animals which lay eggs, inside which the young ones will develop. The animals which give birth to young ones are called viviparous animals. Example, human beings, dogs, cats, etc. Those animals which lay eggs are called oviparous animals. We know that the young ones of lions, hens, human beings resemble the parent, but there are some animals whose young ones do not resemble the parent. Let us take an example of a frog. The life cycle of a frog consists of different stages. The first stage of the frog's life cycle is the egg. The egg hatches to form the larval stage called the tadpole. The tadpole consists of fins and tail, just like fish. During the growth phase, the tadpole grows into an adult, during which it loses its tails and fins. This transformation of a larva into an adult through drastic changes is called metamorphosis. In organisms like a hydra, there may be one or more bulges on the parent body. These bulges are the developing new individuals and they are called buds. In hydra, the new individuals develop as outgrowths from a single parent. This type of reproduction in which only a single parent is involved is called asexual reproduction. As the new individuals develop from the buds in a hydra, this type of asexual reproduction is called budding. Another method of asexual reproduction is observed in amoeba. Amoeba, as you have studied, is a single-celled organism which begins its process of reproduction by the division of its nucleus into two nuclei. This is followed by the division of its body into two, each part receiving a nucleus. Finally, two individuals are produced from one parent amoeba. This type of asexual reproduction, in which an animal reproduces by dividing into two individuals, is called binary fission. Now let us see what is cloning. Cloning is the production of an exact copy of a cell, any other living part, or a complete organism. Cloning of an animal was successfully performed for the first time by Ian Wilmot and his colleagues of the Roslin Institute in Edinburgh, Scotland. They cloned a sheep named Dolly successfully. Dolly was born on 5th July 1996 and was the first mammal to be cloned. During the process of cloning Dolly, a cell was collected from the mammary gland of a female fin Dorset sheep, and an egg was obtained from a Scottish black-faced ewe. The nucleus was removed from the egg. Then, the nucleus of the mammary gland cell from the fin Dorset sheep was inserted into the egg of the Scottish black-faced ewe, whose nucleus had been removed earlier. The egg thus produced was implanted into the Scottish black-faced ewe. Development of this egg followed normally and finally Dolly was born. Though Dolly was given birth by the Scottish black-faced ewe, it was found to be absolutely identical to the fin Dorset sheep, from which the nucleus was taken. Since the nucleus from the egg of the Scottish black-faced ewe was removed, Dolly did not show any characters of the Scottish black-faced ewe. Dolly was a healthy clone of the fin Dorset sheep and produced several offspring of her own through normal sexual means.